Okay, good afternoon, everybody. And this afternoon, we are going to be considering chapter 6, and we're going to be discussing microbial growth, remembering that we've talked extensively now about metabolism. We saw that metabolism was the management of matter, of atoms and molecules for a particular purpose, which was to build them into biomolecules. In doing so, the living organism, the cell, needs to manage energy as well. And it's all to the purpose of growth, of building up biomolecules within the cell so that it can reach a certain size where it can reproduce. Now, we are only going to be talking about the prokaryotes, not about all microbes, remembering that some microbes may be reproducing sexually. We are only going to be talking about the prokaryotes, which reproduce by binary fission. They grow, they reach a certain size, and they divide in two. And that's the process of binary fission. But that places certain constraints upon the principles of growth of whole populations, which we're going to see as we carry on our discussion. So remembering also that there are certain requirements for growth to take place. And first of all, there are a set of physical requirements. The organism needs to have, for maximum growth, needs to have optimal conditions of temperature, pH, and of osmotic pressure. We'll discuss those in some details in a minute. But as I say those things, remember that, especially in the case of temperature and pH, what may be happening is constraints may be placed upon growth because constraints are placed upon the activity of enzymes within the cell. Second of all, the organism requires certain chemical requirements. In other words, it needs the molecules to build biomolecules. If it's growing, it needs to be making extra biomolecules. And for that, it needs things like carbon, for example, which is the backbone of all biomolecules. Second of all, it needs other elements such as nitrogen, sulfur, phosphorus, these kinds of elements as well, which have different purposes, which we'll discuss in a minute. In addition, it may require the organism may require trace elements. If it is an aerobic organism, that is one which uses oxygen, then it will require oxygen for maximum growth. But remembering, as we discussed, that as we have heard before, there are organisms which do not require oxygen, which are able to survive anaerobically. But if they are aerobic, then they will require the presence of oxygen for maximum growth. Lastly, the organism may require organic growth factors, which it cannot itself manufacture. Now, prokaryotes are extremely good at manufacturing everything that they need, but even they may sometimes need external sources of organic growth factors, such as vitamins. Okay, so let's have a look at some of those physical constraints first of all. One of the most important and obviously pervasive is the influence of temperature. And what we find is that populations of organisms, specific organisms, are adapted to specific temperature regimes. And if we look at them, we'll find that they have a particular temperature where they can achieve maximal growth. That is the optimum temperature. We're going to take this group here, first of all. The groups on the populations can be characterized by those temperatures, the optimum temperatures, and are given specific names. So, for example, this group here are the mesophiles. And the mesophiles have an optimum growth at about 37 degrees Celsius, around about there. If we look at a whole population, you may find some species of mesophiles one way, a little bit this way, a little bit that way. But in general, for all of the mesophiles, this is true. They're around about 37 is the optimum. And they have a minimum growth temperature. That is down around 10. and 10 degrees Celsius. And this is at the temperature at which growth will actually stop. Between the optimum and the minimum, growth will slow down and it will stop. Now, it's important to note that usually if we bring them down here and their growth stops and we then raise the temperature again, growth will recommence up to the maximum, up to the optimum. However, if we take an organism past its optimum, like here, and we start raising the temperature when its optimum is at 37.5, very rapidly the growth rate will fall. And the reason for that is, of course, that its enzymes are beginning to be affected. Enzymes, you'll remember, are very much affected by temperature. And we've already seen this kind of curve being the curve of activity that is associated with the impact of temperature on the functioning of enzymes. The point being that in this case, if I take the temperature much higher than the optimum, growth will rapidly decrease until it stops. But in most cases, if I now lower the temperature, growth rate is not restored. And the reason for that is that the enzymes of the organism are denatured at a higher temperature. The actual physical structure of the enzyme, the proteins of the enzymes, is so drastically altered, it is permanently damaged, and we refer to them as being denatured. So this group here, the mesophiles, are important. What else has a temperature around 37.5? Well, that's us. Most mammals, for example, have their temperature around 37.5. They keep a constant body temperature. And in fact, many vertebrates keep their body temperature around 37.5, even if they don't maintain a constant body temperature. They attempt to enter an environment where it is around there. So all of those organisms, many organisms associated with vertebrates will have an optimum around there. And that includes all of our parasites, all of those pathogenic bacteria which are able to infect us. So the mesophiles are a very, very important group. And they also occur free in the environment. And because this temperature regime here is approximately the temperature regime that you would see in the surface of soils, in the atmosphere, in close to the earth, and in surface waters as well. So these are the mesophiles. Meso, meaning they like the middle. Mesophile, liking the middle. Let's take another group, and we're going to go to the other end of the spectrum and realize that some organisms are adapted to other environments. Remembering what I've told you before, that prokaryotes occupy virtually every ecological niche on the planet. They also occupy niches which are extremely cold. And those organisms are specially adapted to the cold. Look here at their optimum temperature. It's right down around about 11 or 12 degrees Celsius. So this would be the case, for example, for many of the oceanic waters, and especially the deeper waters, and those approaching the poles, those kind of places. Organisms there are adapted to the cold. Their enzymes and everything are structured so that they have an optimum around about 11 or 12 degrees Celsius. Again, they have a minimum. This is very, very low. They can continue growing to a very low temperature, but their maximum, their upper limit, is also relatively low. This is only about 18 degrees Celsius, which an 18 degrees Celsius day, you've already got a sweater on. So, and that would be regarded as a temperature at which their enzymes would begin to denature. The psychrophiles have a positive requirement for cold. The psychotrophs here, again, very, very often oceanic and freshwater organisms, They are adapted to cold, but not as great as it would be in really deep waters or in Arctic waters, those kinds of environments. Their optimum is around about 20, 22, 23, somewhere around about there. And again, maximum around about 30 degrees Celsius. So these would be these are able to tolerate cold. They like cooler temperatures. And this would be the case for many environmental bacteria, that they would have an optimum around about there. There is other, however, strange groups which can tolerate high temperatures. This group here, called the thermophiles. Thermophiles have a requirement for high temperatures, and you can see why, because their optimum is there, and that's all the way up around 62 degrees Celsius. That is really hot. To give you an idea, if you have a bottle that's filled with liquid that is at 62 degrees, you can hold it briefly, but you can't pick it up. You can't hold it sufficiently long to actually pick it up. You would want to hold it and then let go. It would be too hot for you to hold. And yet they function perfectly well at that temperature. They have a maximum over 70. That would actually burn you. 72 degrees Celsius would actually burn you. They do wink out at that temperature, but that is an extraordinary range of temperatures to be operating at. 40 degrees is their minimum. That's already really warm. And then the optimum over 60. Where would you find those kinds of organisms? Well, this would be in places, for example, like hot springs, like thermal springs in Yellowstone, those kinds of specialized environments. But there's another group who are even more extraordinary. I want to point something out to you, and that is that around about this temperature here, strange things begin to happen to DNA. DNA, you know, is a double-stranded molecule, and at these higher temperatures, around 70 degrees Celsius, the DNA will begin to melt. The two strands of the DNA will separate from one another in most organisms. But in these organisms here, the hyperthermophiles, they have a very broad range, optimum range. Their optimum goes from 85, which is extremely hot environment rather. These are mostly aquatic, so I can say it's hot water. From 85 all the way to over 100. That is beyond boiling point. There are no other vegetative cells that will survive beyond boiling point. They must have very, very specialized enzymes, and they must somehow be able to protect their DNA from melting at these high temperatures. Otherwise, it simply wouldn't function. So these are the hyperthermophiles, and they have a very specialized metabolism, very specialized structure that allows them to survive these extraordinarily high temperatures above boiling point. Well, where on the planet are you going to find water that is above boiling point? Normally, if you reach 100 degrees Celsius, you can't raise the temperature anymore. If you think back to your physics, at atmospheric pressure, if you raise water to 100 degrees Celsius, it boils and you cannot raise the temperature anymore. You have to, in fact, raise pressure in order to be able to raise the temperature of water above 100 degrees Celsius. And these hyperthermophiles occur in specialized circumstances, for example, in the very deep ocean at volcanic vents, where the pressure is so great that the water can be heated well above boiling point, and they are able to survive up to 110 degrees Celsius, which would be lethally hot for any other organism, including us. So this has implications in practical terms for us, these temperature requirements and regimes, because many of, for example, of the spoilage organisms that we deal with, food spoilage, food poisoning organisms, pathogens, and things like that that can be transported by food, are actually mesophiles. And look what happened here. They're optimum around 37. If we drop the temperature, we can actually stop their growth entirely around about 10 degrees Celsius. So this gives us the chance to actually be able to maintain foodstuffs and things like that at a low temperature and keep them out of what we regard as a danger zone. This is regarded as the danger zone because it runs from about 15 degrees Celsius to about 50. And this is the range where many prokaryotes are able to grow very, very rapidly. If we drop that below that about 10 degrees Celsius, this is where we start to reach refrigerator temperatures. And this is why a refrigerator works. It lowers the temperature so that we lower the environment below, well below the optimum, down to this minimum requirement here. So the bacteria can't grow. Only these, the psychrotrophs, would be able to grow at that temperature. You unlikely find psychrophiles, but there are a few, I have to tell you, a few psychrotrophs that we do have to worry about. And one of them is called Listeria. And Listeria is a common food poisoning, serious food poisoning organism. And they are actually able to grow at refrigerator temperatures. So that would be one that we'd have to worry about. But most of the disease-causing organisms, if we keep them at this temperature, we can stop their growth. So there is one other thing I want to point out, and that is we're talking here about what we call vegetative cells. Those are actively growing, metabolizing cells. We're talking about raising and lowering their temperatures. For most vegetative cells, which are mesophiles and below, if we raise the temperature up to about 60, then the temperature actually becomes lethal to a vegetative cell. It destroys most cells' proteins. So that is fine. This means that we can use high temperatures around about 60 to destroy most cells. But there is an exception to this. And that exception is that organisms which produce spores, members of the two genera, Bacillus and Clostridium, which are able to produce spores, their spores are extremely resistant to heating. And you can heat them up to 100 degrees Celsius in boiling water, and they will still survive. That's a great problem, because if we are trying to boil things to sterilize them and we have spores present, we can't sterilize them. Instead, what we have to do is utilize that same principle that we talked about before, that if we raise the pressure, we are able to raise the temperature of the liquid above 100 degrees. We can raise above boiling temperature. And in fact, we can raise up, if we raise up to about this range here, 120 to 130, we would be able to kill off spores as well. And that is the principle of the autoclave. So the autoclave is a machine which is basically a pressure cooker, raises the pressure. You can raise the temperature of fluids in there above 100 degrees to about 120, keep them for 20 minutes or so at that temperature, and kill off all living things, including spores. Okay, so the next really important physical constraint is that of pH. Remembering that pH is a measure of the amount of protons that you have in a liquid. It is the proportion of protons that are present. And a high proportion of protons implies that you have an acid. A low proportion of protons implies that you have a higher proportion of hydroxyl ions, OH minus, and the pH rises. So an acid has a low pH, and an alkali solution has a high pH. Most prokaryotes prefer a relatively neutral pH, around about 6.5 or 7.5. But there are other organisms. Molds, for example, tend to prefer a slightly acidic environment, not markedly, but slightly more acidic, 5 to 6. There are specialized organisms which grow in extremely acid solutions, and these are called the acidophiles. And there are some which are even alkalophiles, and these adaptations to extraordinary environments are very often the hallmark of Archaean organisms, the other prokaryote group that we seldom talk about. So a neutrophile has its optimum pH around about 7. What is constraining the pH requirement? Again, it's the functioning of the proteinaceous enzymes, because pH affects the structure, the physical structure of the proteins that comprise the enzymes. Think back to your biology classes and you'll remember that proteins are very long molecules. They are polymers of amino acids. They're essentially linear molecules which fold themselves into very specific shapes. And those shapes are maintained by certain kinds of bonds. And the most frequent bond that determines protein structure is a hydrogen bond. And hydrogen bonds are very much affected by pH. So if you start raising or lowering the pH of a solution, you will start affecting the structure, the physical structure of the proteins by disturbing the hydrogen bonds that hold them in place. So the vast majority of organisms that we deal with, especially in health situations, are neutrophiles. They are not acidophiles or alkalophiles. These are relatively exceptional environmental situations where we might find specially adapted prokaryotes. So here is the next physical constraint on organisms for growth. And this is the question of the osmotic pressure which is being exerted upon the cell itself. Now you need to remember the cell is defined by its cell membrane, and that membrane is semi-permeable. It will allow the passage of certain things like water, but it will not allow the passage of certain other things like ions and salts and those sort of things, except through specially controlled passageways through the membrane. So here's the way to conceive of the whole importance of osmotic pressure. If you need to always think about one side of the membrane in relationship to the other. So here we'll use a red blood corpuscle as an example. The red blood corpuscle is a cell defined by its cell membrane. And on the inside, under normal circumstances, on the inside, there is a very high concentration of salts, of biomolecules, of all sorts of things that physically exclude water. So in other words, when we think about it, the concentration of water on the inside of the cell under normal circumstances for most cells, there is less water on the inside usually than on the outside. Now, there are circumstances, such as this one described here, where the cell may be in what is referred to as an isotonic situation. That is, there is as much water outside as there is inside. In other words, there are as many solutes and everything outside as there are inside the cell. The water concentration inside the cell in an isotonic situation is the same as that outside. So now let's just conceive of one water molecule. So a one water molecule now makes its way across the membrane to the interior of our red blood corpuscle. What happens? Well, what situation have we set up by doing that? The inside of the blood corpuscle is now one molecule of water more concentrated than the outside. And what has to happen? One water molecule has to leave. And that is exactly what happens in an isotonic situation. It's not that no water moves across the membrane at all. It's as much water moves across the membrane will have to move out. Otherwise, the isotonic balance is disturbed. So this movement of water back and forth across a membrane, what is driving it? Well, it's a diffusive property. And that is the diffusive property of trying to move a substance from an area of high concentration, in other words, high water concentration, to an area of low water concentration. And that movement of water, the diffusive movement of water, is specifically referred to as osmosis. And that movement of water is an osmotic pressure. So now let's move away from the isotonic situation, which is easy to understand, and instead have a look at the other kinds of situations where the amount of the concentration of water outside and inside may be radically different. This is a situation here which is highly specialized in actual fact in nature. For example, in our blood, we try to maintain an isotonic situation, and that is the responsibility actually of our kidneys to do so, so that this red blood corpuscle does not have strains placed on it by osmotic pressure. It is kept in an isotonic situation. But let's take the same red blood corpuscle, and we're now going to move it into water, into an aqueous solution which has a very low concentration of solutes, has a low concentration of salts and everything else. If that is true, then it has a very high concentration of water molecules, right? It has a high water concentration out here, out this side of the membrane. And on the inside, which is the cytoplasm, which is packed with biomolecules, packed with salts of all sorts, which guide the function of the cell, there is a lower water concentration. Now what happens? Water molecules are going to move by osmosis, by the diffusion of water, into the cell. And the consequence is that that red blood corpuscle is going to begin to swell. And it'll swell actually until it bursts. This is very easy to demonstrate experimentally, because the red blood corpuscles, because they normally occur in a relatively isotonic situation, they have no mechanisms for pumping water out. Instead, water flows in passively through osmosis, driven by the osmotic pressure, until the cell bursts. Now, we have heard about this before, because in prokaryotes, this is a constant danger. Water, in most situations, water tends to flow into the prokaryote cell across the membrane, and they too have no mechanism to pump water out. Instead, the cell begins to swell, but in the prokaryotes, the cell swells up against the cell wall, which is entirely different to the cell membrane. It is a mechanical structure that the cell membrane swells up against. The inside of the cell becomes pressurized. We refer to that as the turgor pressure. And when the turgor pressure is the same as the osmotic pressure, the turgor pressure is the same as the osmotic pressure driving water in, then the swelling stops, because as much water flows in as flows out. When turgor pressure is the same as osmotic pressure, as many water molecules as flow in through osmosis must flow out of the cell. So now let's take another situation. We're going to take an entirely different situation. We're going to take our red blood corpuscle and we're going to put it into a solution which is very, very high in salts. In other words, it's very low in water molecules. Now the inside of our cell has a higher water concentration than the outside. And the tendency now is for water to flood out of the cell into the environment. And as a result, the cell begins to shrivel. And this is also a very dangerous situation for the cell. Should this occur, there are not many cellular mechanisms to protect the cell, and ultimately the cell will collapse completely and ultimately die. Now, we look at how we are describing these situations. We're saying here is that we've placed the cell into an isotonic solution. The tonicity here refers to the amount of osmotic pressure that is this is going to result in one direction or the other. We always refer to the specific solution that we're talking about. In this situation here, where the outside had a much higher water concentration, we say that this is a hypotonic solution, and the inside of the cell is hypertonic to the outside. We always have flow from hypo to hyper. Excuse me. Same situation here. We refer to the tonicity in terms of the environments we're talking about, outside and inside. Here we say we have a hypertonic solution, and the interior is relatively hypotonic to the outside. Water flows from hypotonic to hypertonic. It flows out. Now, these can be real situations. These can be real situations in nature. And in general, let me just go back here. Organisms must be adapted to these kinds of osmotic pressures. Many organisms, for instance, the protozoa, things like that, the eukaryotes, have ways of pumping in a hypotonic solution like this, which is the situation, for example, in fresh waters, in soils, in these kinds of environments. Organisms must be able to deal with this. They very often have pumps, active pumps that pump water out. These hypertonic solutions are relatively uncommon, but may arise as a result, for example, of dehydration. So, for example, if we place cells in very dry circumstances where water tends to evaporate very rapidly, then that ends up effectively being a situation where the outside is hypertonic. So in the isotonic solution, they're absolutely fine. As much water flows in as out. In a situation where we place a prokaryote in a hypotonic solution, they have the adaptation of having a cell wall, which prevents the bursting. But in a hypotonic situation, a cell placed into a hypertonic solution, so the inside is hypotonic, they have very little adaptations that allow them to survive that. And if we observe a prokaryote cell in that situation, water flows out to such an extent that the cell shrivels away from the cell wall. Here is the cell wall. Here is the cell membrane. The cytoplasm is shriveling, and this may well end up killing this cell, or at least stopping it from growing. So I labor this point because this leads to a very important principle for human beings. This is a way in which we can actually preserve food. We can preserve food if we limit the growth of microorganisms in it. And there are various ways that we can do this, but I'd say there are three common ways of doing so. The first is simply to dry food. Well, if you dry food and you dry the whole environment, this is the situation that you create. You force microorganisms in that food to plasmolyze like this, because all of the cellular water evaporates and the cells shrivel and die. The second way you can do it is by raising the solute concentration outside very, very high. And you can do that, first of all, by salting food, for example. That's why salting food works, to preserve it. It prevents microorganisms from growing because they all plasmolyze like this. The third way is one that you might not immediately think of, but if you think of jams and jellies, very, very sweet, sugary environments, we're doing exactly the same thing. Organisms cannot survive in those highly concentrated sugary solutions because the outside is hypertonic to the inside of the cells, and they plasmolyze and die. And that is why, for us, this is a very important principle. Okay. But I always tell you that you can bet that there are always prokaryotes, at least, which are adapted to extraordinary situations. And there are organisms which are adapted to these very, very high solute, hypertonic environments. And so, for example, you can go to salt pans and find organisms which are happily growing in extraordinarily highly saline environments. And these are the extreme halophiles. And the extreme halophiles, again, these are very often, not always, but very often archaean organisms, which are either able to survive those environments or which positively require them. And this is often true of salt pans and evaporative saline solutions like that. All right, so let's talk a little bit about some of the chemical requirements for growth. First of all, carbon, you will understand. It's the structural backbone of organic molecules. And in addition, the organisms like us who are chemoheterotrophs, we use already created organic molecules as a source of energy, where autotrophs use a different source of carbon. They use carbon dioxide as their source of carbon, and they use the sunlight's energy. But chemoheterotrophs use organic molecules both as the source of energy, but also as their source of carbon to build other biomolecules. The next most important element is nitrogen. And that is because nitrogen forms a component of so many different biomolecules, such as the proteins, DNA, ATP. Proteins because it is an integral part of amino acids, the monomers that make up the polymers, which are proteins. DNA because nitrogen is a part of the nucleotides. And then ATP, which you have already seen. Now go back to ATP, adenosine triphosphate. Look at that picture of it, and you'll see that attached to a central sugar ring, they're the phosphates that we're really interested in, but also attached to that sugar ring is a complicated ring structure, which is referred to as a base. And that is nitrogen rich, and it is a base because nitrogen is in fact extremely electron negative. And it lends it that ring structure a relatively basic structure, a basic nature, rather. Now, where organisms get their nitrogen from is actually an extremely complicated story, and it's one that links all organisms on the planet together in a gigantic global nitrogen cycle. So think of it this way. The amount of nitrogen on the planet is actually fixed. We don't have a source of nitrogen from outside. And so that the nitrogen that we have on the planet has to be continually shared amongst all living things, because all living things require nitrogen. So first of all, it's an important principle that most bacteria, most prokaryotes, to obtain their nitrogen, they will degrade protein, already structured protein material. They decompose it. They take dead material, and they break it down. They break down its proteins and use the nitrogen in the proteins as their nitrogen source for various ends. But the principle is that most bacteria tend to decompose already created biomolecules. And as a result, in the end, they end up reducing the nitrogen ultimately to free atmospheric nitrogen. It's a very complicated cycle, which we may talk about later on. But just accept for the moment that the fate of most nitrogen structured into protein is that it gets turned into gaseous nitrogen species, which then go up into the atmosphere. And there's a big problem. Once you create free nitrogen, or N2, that is extremely non-reactive. It is very, very difficult to again grab that nitrogen and build it into biomolecules. Instead, we have to rely on a group of bacteria which are able to take nitrogen from the atmosphere and fix it. They actually fix it into compounds like ammonia, first of all, or a nitrate species of some sort. And other organisms are then able to use those molecules, those compounds, as their source of nitrogen. But without these bacteria, which can fix nitrogen, the entire nitrogen cycle would break down, and all nitrogen ultimately would end up in the atmosphere as non-reactive nitrogen. So it's thanks to them that we all continue to exist. Other elements, sulfur, occurs in many different biomolecules. Very important in amino acids and in proteins, because it allows proteins, for example, to form sulfur bonds, which structure the protein, establish the tertiary structure of proteins. Other biomolecules such as thiamine, biotin, all of these kind of organic cofactors, these all contain sulfur as well. And the sulfur may also be used in strange ways by prokaryotes. They, in general, if they require sulfur, they will decompose proteins to acquire the sulfur. But there are prokaryotes which can use free sulfur, but can also use sulfur compounds such as sulfate and hydrogen sulfide as their source of sulfur. But remembering, we heard in metabolism that there are prokaryotes which are also able to use sulfur compounds as a source of energy. And that may be something you want to go back and have a look at again. Lastly is phosphorus. And phosphorus, obviously, should immediately spring to mind that one of our most important uses for phosphorus is in ATP, the most commonly used biological useful form of energy is the phosphate bond in ATP. But phosphorus is also used in DNA, RNA, and other compounds as well. For example, it is part of the cell membranes because the cell membrane is made up of phospholipids. They are fatty acid tails attached to a phosphate-rich head. In addition, there may be organic growth factors, which prokaryotes require from the environment. As I told you at the beginning, prokaryotes are very, very good at making all of the compounds that they require, but there are a few which they are unable to make. And they may require these from the environment, or from nutrients. And these are referred to as the essentials, so the essential growth factors. And they include most commonly the vitamins, but may also be amino acids, may also be purines and pyrimidines. Those we'll encounter when we talk about the nucleotides. Lastly, especially for determining enzyme function, the organism may require trace elements. And these are inorganic elements required usually in very, very small amounts. And they, in general, function as enzyme cofactors. Iron, the most important one, being in the functioning of the hemoglobin. Copper, molybdenum, zinc, and some other very strange cofactors may occur as well. All right. I'm sorry to do that. I'm just trying to do. Oh, no, that's not what I wanted to do. Oh. I'm sorry. Just let me just stop there for the moment because I've actually done something wrong. I'll get back to you right now. All right, I've done entirely the wrong thing. So we're just going to do it without my face sitting staring at you. Hold on, maybe I can do it. Oh, sorry for dithering around. I did something really stupid. And everything kind of went on the fritz. Okay, so let's just have a look at the impact, especially of this really important atom, which is oxygen. And so let's just remember a couple of things about oxygen. I have told you this before, but it bears repeating, that life evolved on a planet which was entirely free of free oxygen. There's plenty of oxygen, but it was bound up with other atoms, like in water, for example, and in metal oxides. All those sort of compounds may have contained oxygen, but there was no free oxygen in the atmosphere. And it was the group of prokaryotes, which were the cyanobacteria, which evolved the ability to take simple carbon dioxide, water, the sun's energy, put it together to form complex biological molecules, which are the sugars that could then be used to build everything else. But in the process of photosynthesis, they released oxygen. And they released so much oxygen that it began to build up in the atmosphere. Now, you realize that no organisms living then had ever had to deal with oxygen before. And this caused a huge crisis on the planet because the vast majority of living prokaryotes, which was all that there was of them at that time, were unable to survive in the presence of oxygen. And the reason for that is that oxygen is an extremely reactive molecule. It is very, very electron negative, and it is extremely reactive, and it literally burns through biological molecules. In addition, it exists in several different forms. And one of those forms is as a superoxide, which is a negatively charged oxygen ion. And it's actually O2 minus. And it's very often depicted chemically as HO2 minus, not H2O, but HO2 minus. And it's called the superoxide. And it's even more reactive than oxygen on its own. And that proved deadly for the vast majority of living prokaryotes. So most of them went extinct, but a few were able to do a couple of things to evade the impact of oxygen. The first was to evolve a set of enzymes which could deal with the superoxides. And the most important of those enzymes still exists today to do exactly that. And it is called superoxide dismutase. And here it is here, written out here, superoxide dismutase, and that is SOD. Now, SOD, superoxide dismutase, converts that superoxide to another compound, which is called peroxide. Now, the peroxides are also extremely reactive. That's why we can use them as disinfectants. And organisms evolved another enzyme called catalase, which breaks down breaks that down to just water and to free oxygen again. And the presence of those two enzymes enable most organisms that have them to withstand the presence of free oxygen. The other great innovation to deal with free oxygen was the evolution of cellular respiration, which originally, we believe, evolved to actually take that free oxygen and to combine it with hydrogen, in other words, reduce it to water, which is harmless. So you understand that the presence of free oxygen drove substantial evolutionary steps in prokaryote metabolism, which then was inherited by everything that evolved from those prokaryotes. So oxygen had an enormous impact on the history of the planet and on the evolution of metabolism in general. But its impact on cells still remains. And its cells still have to deal with the presence of oxygen, and they have different strategies for doing so. So first of all, let's just picture an experiment. What we're going to do is, accepting that we have about 20% free oxygen in the atmosphere, so we've got a lot of free oxygen in the atmosphere, we're going to set up an experiment where we're going to take a tube of agar, and we're going to heat it. We're going to boil it to drive off all the oxygen that's present in it, and we let it solidify. And then we're going to inoculate it with different bacteria and see how they behave. If we take a tube of agar like that and just leave it, what we'll find is that at the very surface, ignore the presence of organisms here. At the very surface here, where it's in contact with the air, we'll have a high level of oxygen. At the very bottom here, we'll have no oxygen. We say that that is anaerobic. Down there, it lacks oxygen entirely. In the middle here, there would be a gradation from a lot to very, very little. So now let's put different organisms in there and see what they do. So this first organism, we inoculate the whole tube, we'll stab right down to the bottom. We'll come back a day or two later, and what we see is it only grows at the very top. In other words, it has a positive requirement for the presence of oxygen in order to grow. And in order to do that, it has to have the requisite enzymes for dealing with oxygen. And those are catalase and superoxide dismutase. And this allows toxic forms of oxygen that are naturally present, but which also arise, in this case, from cellular respiration. Cellular respiration itself may produce toxic forms of oxygen, and in particular may produce peroxides, H2O2, which requires even more catalase to be present. So these organisms possess all the necessary mechanisms to withstand the presence of oxygen, but they have a positive requirement. They're entirely dependent on cellular respiration. They can only grow up here in the presence of high levels of oxygen. They can't grow down here where there is no oxygen. Look at this organism here, the different organism inoculated into the same kind of tube. And what we see is there is growth throughout the tube. It's still able to grow under low oxygen and in the absence of oxygen. But it grows most abundantly where there is oxygen present. This is a facultative anaerobe. That means it is able to survive the absence of oxygen, but it prefers the presence of oxygen. Why? Because in the presence of oxygen, it is able to produce abundant ATP from cellular respiration. Down here, where there is little to no oxygen, it is entirely dependent on glycolysis for its ATP. And that is a very inefficient way of producing ATP. So growth here continues, but it is slow. Growth in the presence of oxygen is fast and abundant. This organism must also be able to produce catalase and superoxide dismutase to deal with the high levels of oxygen up here. So here's our third organism. Again, same tube, same kind of tube. We inoculate it and we see that it can only grow down here at the very bottom. And this is a facultative anaerobe, not facultative, an obligate anaerobe. This is a facultative anaerobe. This is an obligate anaerobe. That means it has an absolute requirement that there be no oxygen present. Many of these obligate anaerobes cannot tolerate even a few atoms of oxygen in their vicinity before they die. So they need an absolute absence of oxygen, and they lack any of these enzymes. They don't have catalase, they don't have superoxide dismutase. There are several of these obligate anaerobes. The most important one, probably, that we'll hear about is a genus called Clostridium. And we'll talk about it at some length when we talk about disease process. So there are a couple of other sort of strange situations. This organism here, we stabbed it into the same kind of tube, but look what happens. It grows throughout, just like the facultative anaerobe, but it does not grow better in the presence of oxygen. And this is because this organism it has a superoxide dismutase. But it can only partially inactivate the harmful forms of oxygen. And it has no cellular respiration. And as a result, it grows slowly throughout, and it is growing slowly because it is entirely dependent upon glycolysis and fermentation processes. Even though it grows up here, it's just tolerant of the presence of oxygen. Its growth doesn't improve in the presence of oxygen. So this is an aerotolerant anaerobe. Its growth is anaerobic even when oxygen is present. This group here, there are some very important pathogens in this group. And this is these are the microaerophiles. And look where they grow. They grow at low oxygen concentrations. And they are using aerobic growth. They're using cellular respiration, but they can only do so at a low level. And they are unable to withstand the toxic forms of oxygen that occur at very high levels. These are the microaerophiles. Okay, so there was one more. Just give me one second. I'm sorry to do the round. So, I thought, in any case, it doesn't matter. I think I'll get to it later. There was one other that I wanted to mention. Okay, so here's another constraint on growth, which I'm sure you've not heard about, because this was only discovered relatively recently. And this is a fact that full bacterial growth cannot occur until bacteria actually talk to one another and establish that conditions are correct for growth. And this is a system called quorum sensing. Bacteria actually release molecules called inducer molecules, which other bacteria pick up. And until the level of inducer molecules reaches a certain level, bacterial growth will not proceed at optimum levels. So this is another constraint on bacterial growth. There have to be other bacteria present. And it's a way of signaling that, in fact, conditions are correct. Conditions are ideal for growth to occur. And this is especially important in establishing microbial communities. Now, microbial community is a mixture of different kinds of microorganisms. And these are tremendously important because many microorganisms only grow well, first of all, when they're attached to a surface, and second of all, when they are growing in community. And this enables them to share nutrients, and they build an actual structure by linking the glycocalyx, for example, really importantly, linking their glycocalyx structures into a gel-like structure or slime. And this forms a biofilm. And biofilms are really important, especially in health situations. They're important throughout nature, but they become very important in certain clinical situations as well. For example, we have biofilms which coat our whole intestine. There are biofilms which coat most surfaces in aquatic systems. And in the establishment of this biofilm does all sorts of things. First of all, it allows, they're structured so that water can actually flow through them, but it protects these the microorganisms from external threats, like predation, for example, toxic substances, whatever. In addition, it forms clumps which can break off, float away, and seed another community somewhere else. And the reason I said that this is important clinically is because these biofilms very often form on the surfaces of artificial pipes and tubes and things like that. And because of the structure, the density of the biofilm, they're often resistant to disinfectants and things like that. Now, this becomes extremely dangerous in a clinical setting because these biofilms can form on the interior, for example, of catheters, indwelling catheters. They form very rapidly on contact lenses. These biofilms are primarily responsible for dental caries, which is the decay of teeth. And in hospital settings, biofilms are involved in about 70% of infections encountered in hospital settings, or nosocomial infections. So they are tremendously important. Here's a typical biofilm, and this is an organism which grows quite rapidly and readily in urinary catheters. And this is Serratia, growing on a plastic catheter. And you can see how it's attached. The biofilm is attached by capsular structure, and they are bound together to one another by those same kinds of structures. All right, let's talk a little bit about the culture media, about growing bacteria in a laboratory setting. You will have already encountered many of these things in lab, whether you understood clearly that that's what you were seeing or not. You will have seen some of these principles in action in the lab. Now, first of all, a culture medium is a nutrient medium which is prepared specifically for microbial growth. Excuse me. There are two principal forms of that. And the first is in the form of, hold on one second. Excuse me. The first is in the form of simply a solution of nutrients, and that is a broth. And a solid medium is essentially a broth into which we put some sort of solidifying agent. And that is usually agar. Not always. We can use some other solidifying agents. But we usually use agar because it's so readily available and it's such a standard procedure. And it has advantages that I'll discuss in a minute. We usually start culture with a sterile medium, either broth or solid, and that is one that has no microbes present, and that is usually a medium which has been autoclaved. That is kept above 120 degrees Celsius in a pressure cooker system called the autoclave for about 20 minutes. An inoculum is what you introduce into the medium in order to form a culture. And the culture is the resulting healthy growth of the culture of the organisms that you wanted to culture. So there are two kinds of medium that are commonly encountered in the lab. The first is a chemically defined medium. And in a chemically defined medium, we know the exact chemical composition and we know the exact chemical concentrations of every component. So they are very often constructed to grow what we call fastidious organisms. And those are organisms which require very particular growth factors and growth conditions. The chemically defined media are not ones you are going to encounter in your lab practice. They're very often used, for example, to study nutrient requirements. So we can, for example, create a chemically defined medium, and then also one of the components, the concentration of one of the components, to see the impact on growth of organisms. That would be a simple experiment using a chemically defined medium. The media that you generally encounter in lab and most commonly used in labs are called complex media. And these, although they are made to a strict recipe, we don't know the exact composition or the exact concentration of all of the components. So, for example, they usually depend on extracts and digests of meat, plant material, or yeast, or whatever. But the actual chemical composition, we don't know. And so, for example, if we have a nutrient broth, we usually use a yeast extract. Well, yeast extract, we may add, it's precisely 1 gram, but we don't know exactly what's in the yeast extract. That is usually done. Yeast extract is usually used because it's a rich source of growth factors. It does provide some other stuff as well. But then we provide a rich amino acid source, which is usually a digest of meat or something similar, some similar protein source. And you'll see that these are often called peptones. A peptone is a meat which has been digested with pepsin, which is a stomach enzyme. And a tryptone is also a meat which has been digested with a pancreatic enzyme called trypsin. So just, I'm sorry to do this. I've got something stuck in my throat. And as I said before, a nutrient broth is simply a solution. A nutrient agar is a nutrient broth that is solidified, usually with agar. And this, we should just mention a couple of the characteristics of agar. Agar is a very complex polysaccharide. It's extracted from seaweed. You would have heard the reason for that, for the importance of that, is very few bacteria that we deal with, pathogens or whatever, have ever seen agar in the natural world. So they don't have the enzymes necessary to digest it. So it stays solid. And its really important characteristic is that it liquefies at 100 degrees Celsius. So you boil it and it liquefies completely. But when you cool it down, it solidifies between 40 degrees Celsius and 50. Now that is very, very useful because it means it stays liquid for a long time. And it also solidifies at a temperature where many bacteria will actually survive. If we want to make a suspension of bacteria, for example, which we do when we are using certain counting techniques, which you'll hear about later, that we can suspend them in cool agar immediately before it reaches setting temperature and still pour our media. So here is a typical nutrient agar, and this is just a complex medium. This is exactly what you would see in the lab. These are the sorts of places that you're dealing with. First of all, there's a peptone. Now that is a digested meat, it's a protein, which has been digested with pepsin. We know exactly how much peptone we're going to put in, but we don't know the exact amino acid composition of this. We just know there's way more than we need. That's all. We know that there's way more of all of the amino acids than we would possibly need. We add something like a beef extract or very often yeast extract as a source of growth factors, vitamins, things like that. We may add salt like sodium chloride or others if we want to ensure that, for example, we establish in isotonic conditions or something like that. This is not always necessary. It depends on what we're doing. Then we add the standard amount of agar, which is 15 grams of agar to a liter of water. And that is almost invariable, 15 grams to a liter. is sufficient to create the kinds of solid media that you've seen already in lab. Here is a chemically defined medium, on the other hand. And this is looking at to do nutrient experiments, for example, on E. coli, something similar to that. And here, I'm not going to belabor the details here. All I want to show you is we know precisely the amount of every chemical that is involved in creating that medium. And you may look at this and say, but where are the proteins or the vitamins or everything else? The answer is, in this case, you don't need to add them, because E. coli presented with a sugar, glucose, and a source of nitrogen and phosphate, and sulfur and potassium, is able to create, is able to synthesize everything it needs. It's able to synthesize from these basic components all of its amino acids, all of its proteins, all of its growth factors. So it doesn't require an external source of organic nutrient. It can produce everything from these inorganic components. There are some special media. I think that you may encounter at least one of these. For example, if we want to grow anaerobic bacteria, which cannot tolerate the presence of oxygen in order for them to grow, we can do so. We can create a special medium, for example, using a compound like sodium thioglycolate, which will combine with all the free oxygen and deplete it in the medium. And what we will usually do, we'll make the medium and then heat it up, drive off all free oxygen, then cool it down and inoculate. And then as the bacteria grow, the sodium thioglycolate will ensure that no free oxygen diffuses into the medium to kill off the obligate anaerobes. You can also grow anaerobic bacteria in an anaerobe chamber of some sort. This is an anaerobe jar, and it's a simple thing, a very tightly sealing lid, together with a set of compounds and catalysts, which will combine all of the oxygen, free oxygen inside the jar, and convert it to carbon dioxide. And it usually fills the jar with hydrogen as well, to create a totally anaerobic environment. But there are some anaerobes which are so sensitive to the presence of oxygen that we are absolutely unable to grow them even in this kind of situation, because we cannot ever expose them to the presence of oxygen. And instead, an initial culture may be started from spores. And then once we have living vegetative cells, they are kept in this kind of anaerobe chamber, which has airlocks, which whenever we move anything in or out, we place everything in the airlock first of all, totally exhaust all the oxygen, replace it usually with carbon dioxide, before we move anything into the anaerobe chamber. So the interior of the anaerobe chamber is kept entirely free of oxygen for its entire life. Oh yes, this is the slide I thought I had on earlier. When we were talking about microaerophiles, some of the microaerophiles are also capnophiles. And what that means is that they are organisms which not only require very low oxygen levels, they also require high carbon dioxide levels. Now we can do that by just growing them in a chamber with an envelope, a packet produces carbon dioxide. But there is a very old-fashioned and very simple way of producing a really suitable environment for them, and that is to set up what's called a candle jar. And here's one here. Here are our petri dishes inoculated with our capnophile source, and we then light a candle in the jar and close the lid. The candle will use up a lot of
not all of the free oxygen, but most importantly, it will raise the carbon dioxide levels really high before it goes out. And this is a method which is still used. And I mention it because this is a way of growing one of a really important genera that we will talk about later, which is Neisseria. And there are two Neisserias in healthcare that you should know about. One is Neisseria meningitidis. Neisseria meningitidis is the causative agent of bacterial meningitis and a very, very dangerous organism. The other is Neisseria gonorrhea, which causes this sexual transmitted disease of gonorrhea. Both of them are capnophiles. Okay, let's talk a little bit about the kinds of media you have already seen and will see in your lab work. And certainly these are things you need to know as you move into healthcare. These are the kinds of media that you might encounter if you ever work in a clinical laboratory. So we recognize, broadly speaking, two kinds of media that we would use in the lab. Well, let me say there's three kinds. The first is just generalized media that virtually anything will grow on, and that's kind of for routine work. But then we can also make media which are selective. Those are media which discourage the growth of unwanted organisms and which encourage the growth of organisms that we are interested in. So in some way or another, in these media, we include inhibitors which suppress growth. And we'll talk about that in a little bit of detail. The other kind of media are differential media. And the differential media distinguish, especially in the solid media, they distinguish colonies of different microbes on the same plate by revealing different characteristics of those colonies. But many media that we use in the lab are both selective and differential, and you'll see the examples of that. So first of all, this is blood agar, which is a differential medium. Now, it has no selective ability. Blood agar is a very good generalized medium, but it is differential because it is composed of whole red blood corpuscles, usually sheep's blood. And the medium is made in such a way that the blood corpuscles stay entire in the medium. Then we will plate certain organisms onto it. And very often, the organisms that we plate are able to actually break down to one degree or another the blood corpuscles or the hemoglobin or both from the sheep's blood. And this is revealed by changes in the vicinity of colonies. So have a look here and you'll see these are colonies here that have grown after a streak plate. This is the normal background color to the medium, but you'll see around the colony, there is a halo there. And this is called hemolysis. Hemolysis, literally breaking of blood. So the nature of the hemolysis is what we are interested in because different organisms do it differently. That is why this is differential. It allows us to distinguish between the way in which different organisms perform their hemolysis. So have a look here. Here's a plate, a blood agar plate, which has been streaked with 3 different organisms. I'm going to go to this one first. This is beta hemolysis. Here's the beta sign for you here. And you can see that around this where the bacteria are growing there is completely clear. And this is characteristic of beta hemolysis. In beta hemolysis, the blood corpuscles are completely lysed and the hemoglobin is completely digested, leaving a clear halo around the colonies or the growth. That is typical beta hemolysis. This is alpha hemolysis. It's a bit more difficult to distinguish. But have a look around the colony. Can you see this greenish color? There, also in the middle here, and in the growth itself, there is this greenish deposit. There is no clearing. The reason for that is that alpha hemolytic bacteria do not lyse the blood corpuscles. They only partially digest the hemoglobin, and they produce this greenish byproduct of breaking that down. So these two organisms are releasing enzymes into the medium around them, and those enzymes are doing different things because those enzymes are different. We've immediately been able to distinguish clearly between that growth, the organism that is producing this growth, and the organism that is producing this. That is why it is that is how we distinguish on these discriminatory plates. This last one here is called gamma hemolysis, and it's nothing. It's these organisms are unable to do anything. They can't lyse the blood corpuscles for a start, and they can't break down the hemoglobin. So we have alpha, beta, and gamma hemolysis. This is a very, very important test in looking at pathogenic streptococci. And most of the really nasty streptococci are alpha hemolytic streptococci. But there are some bad betas as well. But it's an important discriminatory test for determining which streptococcus you're actually dealing with in an infection. Okay, so that was differential. We differentiate between different kinds. Here's another kind. And this medium is actually both selective and discriminatory and differential. And I'll explain exactly why. I would say for a start, they say here uninoculated. I'm going to just change that a little bit for the purpose of our discussion. And I'm going to tell you that I inoculated this with E. coli. Okay. So I put E. coli on there. And then I did the Staphylococcus epidermidis and Staphylococcus aureus. And here's the results, and I'll explain why we got those results. First of all, E. coli failed to grow at all. It looks as if it's uninoculated. Staphylococcus epidermidis grew perfectly well, and it produced this white streak, and the background remained much the same. The Staphylococcus aureus, on the other hand, produced this yellowish streak with this yellow, distinctly yellow background. Why? Well, first of all, this is mannitol salts agar. That's its name, and you will see it in lab if you haven't already. And mannitol salts agar, as its name implies, has a high concentration of salt. So high that there's not many prokaryotes that can actually grow under those conditions for the reasons that we discussed earlier in this lecture, due to osmotic pressures. So our E. coli failed to grow because there's too much salt. But Staphylococcus, both these species of Staphylococcus can grow perfectly well. That is how this medium is selective. It is selective for facultative halophiles. They are halotolerant organisms, those which can tolerate a high concentration of salt. Why is Staphylococcus tolerant of high concentrations of salt? Well, just think about some of the common sources of Staphylococcus. Staphylococcus epidermidis. Where do you think Staphylococcus epidermidis grows? It grows on the skin. Our sweat contains a high concentration of salt for a very good reason. It's a very good disinfectant. But there are bacteria, part of our microflora, in fact, which can withstand that. And these, most importantly, are staphylococci. So the medium, the high salt concentration selects for those which can withstand it. In this case, the staphylococci. But also contained in the medium is this, and this is a sugar, mannitol. And mannitol can be fermented by various organisms to produce an acid. The pink color in this medium here is an indicator, phenol red. And when an organism is able to ferment mannitol, it produces a yellow color because it releases acid. So this test is not only selective for Staphylococcus, it is discriminatory or differential as well, because it can tell us the difference between two species of Staphylococcus. Epidermidis is unable to ferment mannitol. There's no yellow color. Aureus is able to ferment mannitol. It produces a yellow color. So this is a differential response by two different species of Staphylococcus. So this medium is both selective and it is differential. Here's another one. This is a very important medium which you will see in lab, but is one of the commonest media which is used in clinical labs because it provides us with very important information, especially about bacteria which are derived from the gut. And you'll see the reason for that when I explain how this medium is constructed. Let's have a look first. What they've done is they're streaking various kinds of bacteria, but most importantly, enteric bacteria, those associated with the gut, such as E. coli, Escherichia coli here. Now, here's an ordinary nutrient agar, totally non-discriminatory, totally non-differential rather, and totally non-selective. And here they streak E. coli to show you, grows perfectly well. Staphylococcus aureus, it grows really well. But here's McConkey's agar. And now when we streak E. coli grows very well, produces this kind of purplish black to black streak. But look at Staphylococcus aureus, it is completely unable to grow. This medium McConkey's is selective. It is very strongly selective for gram-negative bacteria, and especially for gram-negative enteric bacteria. So it would be an important medium to use whenever we're looking for those kinds of bacteria or trying to grow them in the lab. So Staphylococcus is a gram-positive, and it cannot grow. And the reason for that is that there are two compounds which inhibit in this medium, which inhibits the growth of gram-positives. And that is, first of all, there is crystal violet, and that should be familiar to you. It's the same compound that you use to do gram stain. There's crystal violet, and there is also bile salt. Bile salts is exactly what their name implies. These are salts which are extracted from bile, and they are strongly selective for gram-negative gut bacteria. Why? Because our gut bacteria are continually bathed with bile salts. Bile is released into the gut to assist in the digestion of fats. So our gut bacteria are continually exposed to bile salts. Staphylococcus never sees bile salt and is killed by the bile salts. So that is how the bile salts select for this kind of bacterium. But McConkey's is also differential, and a very important differential medium, because it contains the sugar lactose, and not all enteric bacteria are able to ferment lactose. It's an important discriminatory test to tell the difference between bacteria which otherwise look and very often behave in extremely similar ways. And here's an example. This is the same streak. This is E. coli here. And this is a very closely related organism, which is Salmonella enterica. And Salmonella, in many ways, is very similar to E. coli. But it's very important to tell the difference between the two, because many of the Salmonellas are in fact pathogenic, where only very, very few E. colis are pathogenic. And here's the difference. You can see this here is this deep purple color, where Salmonella is not. It's yellow. The reason for that is E. coli is able to ferment lactose, and the fermentation of lactose turns it this deep purple color. In fact, if you look at the E. coli streak like this, very often you'll see it has a beautiful iridescent look that is conferred by the crystal violet indicator that is present in the medium. Salmonella is unable to ferment lactose, so its streak, it grows perfectly well. It's not affected by the bar salts, but it does not ferment lactose. So this is a differential response, which clearly tells us the difference between these two. Okay, just make sure, please, that you understand. This is just for your revision. Make sure you understand the difference between all of these, and you understand the purpose of each of these kinds of media. Okay, so let me just very quickly run over the ways in which we use media to produce pure cultures, which is something you will already have done in lab, but you may not have understood clearly the principle of what you were doing. So a pure culture is one which contains only one species, or sometimes one specific strain of a species that we are interested in. But we know that every cell in that culture is essentially the same. Now, you will have seen, as you in street media, et cetera, in the lab, that you end up producing colonies of cells. And in general, we assume that when we see a single colony, that colony has arisen from a single cell or sometimes a spore, at the very most from a small group of cells. We can't always tell that it definitely was a single cell. For this reason, we often looked, instead of using the word colony, we often use the term colony forming unit. So you see a colony forming, it's certainly in most cases probably formed from a single cell, but it may have formed from a little clump of cells. We can't be exactly sure. The commonest way to produce pure cultures is to produce colonies where we are sure that the colony is of a single type that we are interested in, and then use that colony to secondarily establish cultures on new media. And the commonest way to do that is to use the streak plate method that you have already seen in lab. There are as many streak methods as there are microbiologists. It's like a signature. Everybody develops their own kind of streak pattern, but as long as they work, then that's fine. But I just want to explain a principle here that you may not realize when you did this. What you're actually doing in a streak plate is you actually using a dilution method. It's not a precise dilution method, but nonetheless, it is a dilution method. What you're going to do, what you do in a streak plate is, first of all, you use a source of bacteria. And very often that is a single colony from another plate. Remember, a colony that's just visible to the naked eye, as a rule of thumb, contains about 1 billion cells. So we're going to start off with about 1 billion cells, and we're going to do our first streak. And you can usually just do a close streak back and forth here as the first streak. Then you flame your loop. So what are you doing? When you flame the loop, you're sterilizing. Put away your original plate. You're not going to use it again. This is your total source now of bacteria, because you're going to flame your loop, and now you do your second streak. Here it is here. Just think about it this way. As you move down here in your second streak, at the start here, you're going to leave behind a lot of bacteria. As you move through the streak, you're going to be streaking along fewer and fewer and fewer bacteria. You're going to leave most of them behind. Now you flame your loop again. Now you're going to sample through there. All you're doing is you're sampling a small selection of an already spread out population. Now here's your third streak there. And now you flame your loop. Now you're going to sample a very small amount of this by just streaking through there, spreading them out. And by the time you get through here, you're trailing behind very few bacteria, and you're spreading them far apart from one another. That's what I mean when I say this is a dilution method. As you move through the streaking procedure, you are streaking fewer and fewer and fewer bacteria if you do it properly. In the end, you're streaking so few that you separate single cells out from one another, leave them to incubate for a day or two, and each of them produces a colony, which is derived, hopefully, from one single cell, at the most a few cells in as a colony-forming unit, but all of them virtually or absolutely identical to one another. These are pure colonies. And if you sample just one of these and now do the same thing on a new plate, you've established a pure culture. And that is the ideal, experimental ideal to work from. So let's just discuss finally for today the actual process of bacterial growth and of cell division. So first of all, bacteria are cued to divide by their size. So they grow to a certain requisite size. The cell has ways of monitoring the whole process. At a certain point in the growth, the entire genome will reproduce. So remember that the genome here consists of a single chromosome here, which is circular, and that single chromosome will entirely replicate. And the two replicants are separated from one another by the growth of the cell. They are actually attached to the cell membrane at a point here, and they are pulled apart from one another by the growth of the cell. When the cell reaches a certain size, then the cell will actually divide down the middle by constricting the cell membrane here and grow a new cell membrane there, and by splitting the two by individual cell walls. Now, in most cases, these will split apart from one another, but in chain-forming bacteria, they may remain attached to one another for some time, but they are functionally entirely independent of one another. And the cells separate, and the whole process begins again. These are small cells. These daughter cells are relatively smaller than the original cell, and they begin now to grow to a certain requisite size and repeat the process. What is important about this is that under ideal conditions, this process occurs at a set rate. In other words, if this took 20 minutes, the time for these to do the same thing and divide would be 20 minutes. This can take place extremely rapidly. And in fact, E. coli under ideal conditions will replicate about every 20 minutes or so. It depends on the bacterium you're talking about. Sometimes there are bacteria which reproduce very, very slowly. But in most of the bacteria that we have to be concerned with will reproduce quite rapidly. This has another important implication, and that is from a single cell bacterium, you can in fact end up with an enormous amount of bacteria relatively rapidly. And this is important not only in environmental situations, but especially important in pathogenic and infectious situations, because bacteria, prokaryotes can reproduce very, very rapidly and establish a dangerous condition extremely rapidly that we might have to deal with. This is just an electron micrograph of a dividing cell. The cytoplasm already clearly divided here, one from the other, and now the cell wall busy dividing down the middle here. Okay, so that period of time for reproduction, that is called the generation time, and each division is counted as a generation. And when we look at culture, what we often see is that it's practically synchronized. The cells are really closely timed with one another. So the entire batch will double every generation. And every generation time, under ideal conditions, as which we'll see in a minute, under ideal conditions, that generation's time stays constant. So every time that happens, the number of cells doubles. So if we want to know how many cells are present, what we need to do is we need to take the number 2 and by the power, and that power is the number of generations. So we'll see this now. So first of all, we're going to start off with one cell. 1 is 2 to the 0. If you want to know why it's 2 to the 0, go back to your basic math, go to Google or something, and look up what 3 to the 0 is, or what 4 to the 0 is. They're all 1. It's all 1. So 2 to the 0 is 1 cell. No generation time yet. 1 generation later, 2 to the 1, 2 cells. Another generation, 2 to the 2, 4 cells. 3, 8, 4, 16, 5, 32, etc. You'll realize that we're going to build up enormous numbers very, very quickly. And we're going to, in order to deal with this, we should actually start to use some mathematical tricks. So here we are. Here's the number of generations. We're going to look at something growing for 20 generations. And that is 2 to the 20, 1,048,576, just over a million. And now we're rapidly going to get to numbers which are so enormous that it makes no sense to use the arithmetic computation of 2 to the generation. Instead, it's much easier to start looking at the log to the base 10 of this original number, of the arithmetic number. And if we do that, this is what we end up with. This very, very important series. Because look what happens. Here is a graph drawn of a batch growth of a bacteria. Now, what I mean by batch growth is I've got a flask with rich nutrient in it, and I'm going to start off with a small number of cells. Okay, just drop 1 cell into it if I want to. and I'm going to watch what happens. And this is the ideal growth. We are here, the number of generations, right, along this axis here. And we're going to, as we said here, we're going to go to 20 generations here. And so here's the generations along the bottom axis. Here, we're going to look at, sorry, let's do the side first. We're going to look at the arithmetic number of cells, okay? That's that axis there. Here on this side, we're going to look at the log to the base 10 of this arithmetic number. So what do we see? Well, let's look at the arithmetic growth first. What we see is initially it looks like relatively slow growth. Then it very rapidly increases, extremely rapidly, until when we look at the arithmetic along the axis, it's practically vertical. And that is, after 20 generations, we get to just about 1 million cells there. Remember that this is actually a relatively short time. And that our next generation is going to be 2 million cells, and then 4, et cetera, et cetera, et cetera. We'll soon be in the billions and the trillions of cells. If we take all of the, looking at the arithmetic number, actually soon we just don't get that much information from it. But if we plot the log to the base 10 of that, we see something very interesting, and that is a straight line. And that means that this is logarithmic growth. As we refer to this as logarithmic growth. And when we plot it, we end up with a straight line like this. And this becomes very important and a very, very useful way of computing bacterial population numbers, especially if we know the amount of time it takes for one generation of division to occur, and we know our starting number, we can compute without sampling or anything. We can actually begin to compute the approximate total number of cells that are present in a culture by looking at a graph like this. So remember, this is the arithmetic growth curve there. And this U shape here is typical of a logarithmic growth, which if we plot it as the log to the base 10, we get a straight line. Okay, so what is the implications of this for a real-time situation? So what we're going to do is we're going to do another experiment. This is called a batch experiment. That means I'm going to take a single flask of nutrient. I'm not going to renew the nutrient. I'm not going to do anything. I'm just going to have a batch of nutrient. So in there, I have a set amount of nutrients, obviously. And we're going to follow over time what happens to our bacterial population once we have inoculated. And you'll hear that there are a couple of little complications. First of all, I'm going to drop into that. I'm going to inoculate with a relatively small amount of bacteria. Well, I'm following this curve here, there. And this is a log curve, okay? This is not arithmetic, this is log. So what you're going to see is that for quite some time here, there is very little change. There is, despite the fact that the conditions here are ideal, there's abundant nutrient, we've got it at a good temperature. There's very little competition between bacteria for competing for nutrients or anything like that. And yet, we see very little growth. Why? Well, think back to what I told you earlier in this class about quorum sensing. Quorum sensing, the bacteria talk to one another. And until they're sitting in this ideal conditions, but they are releasing inducer molecules. But there's very few bacteria. So there's very little inducer molecules. Other bacteria are not picking up the inducer molecules. They're not speeding up their growth because they're not getting enough information from the inducer molecules. Only when the concentration of inducer molecules reaches a high enough level, suddenly the bacteria click into full growth. And that occurs here. This period of slow growth is called the lag phase. As soon as they click into proper reproduction, growth and reproduction, this is the period that we saw here. This is the curve that we're looking at here. Because this remember, this is a log scale. This is practically a straight line there. So that's what we're seeing. This is the period of greatest growth. So every generation is doubling. Doubling time, the generation time is a constant throughout this period here. There is abundant nutrient and conditions are ideal. So they just, each generation is faced with the same, pretty much the same conditions as the previous generation. Each of them is doing as well as the previous generation did. And the increase in population size is logarithmic. And as this proceeds, however, conditions are going to change because the amount of nutrient is going to start to drop. In addition, the amount of waste product, and those sort of things, are going to increase. There's going to be competition for things like oxygen, that sort of thing. So the conditions are not going to remain ideal. And that generation time is going to begin to increase. And in fact, we're going to begin to see that a lot of cells are actually not able to make it. And we're going to pass into another stage here called stationary phase. And in stationary phase, the number of cell deaths equals the number of cells that are produced. So we have a straight line here, and that is stationary phase. Remember, this is batch. We're not doing anything to help them out. The conditions get worse and worse and worse. Nutrients become poorer and poorer and poorer. And we enter a phase now when, in fact, the population begins to decrease, and it ends up decreasing at a logarithmic rate. And this is the phase when the number of cells that die cannot be replaced by the number of cells that are accrued from reproduction. And the population begins to crash until ultimately the entire population dies. I will add, though, depending on the organism that you're talking about, in many bacteria, a few cells might survive here, and they're called persister cells. These are very important clinically because these persister cells may, in a clinical setting, be able to seed another infection. If conditions improve for them, they may be able to seed another infection, another population. But I want to return to this here. The stationary phase is very, very interesting phase because all sorts of things happen here. And one of the most important is in a spore-forming organism, for example. This is when spores will begin to be produced. When the nutrient situation begins to become disadvantageous, that is when cells will begin to form spores, which can outlast this entire process until they can find themselves back in this kind of situation if, for instance, they get blown into a favorable nutrient situation. This is a very important curve, and please read up on this in your text and make sure that you understand it. Okay, and I'm going to leave it there for today. There is one more class which I will record. So this week you have these 3 classes on bacterial growth to consider. Okay. I hope that you enjoyed your Columbus Day holiday, and I will see you all next week.